Bloody hell, it's a little longer than I wanted to over that, but I'll keep going. The rest of my time I'm going to spend on new stuff. Some of you know that we relaunched our website recently. This is our new website. It looks nice, but in particular it has lots more content. And the main content that I want to emphasise is the dynamic content. We obviously have a news feed. Everyone has a news feed. That's not very interesting. I mean, obviously it tells you news, but every worthwhile website has one of those. This is the one I want to bring your attention to. Everyone has one of those as well. We have recent news and events and blog posts. But our blog posts are a bit more high-end than most. I get my wonderful research assistant Michael Ray to write most of them. He's the guy who co-authored my book with me, if you remember that. And we don't just take the thing that we saw in the news today and write something in an hour. Michael is a conscientious sort of guy, and he's extraordinarily good. And so you will see stuff there that you might, if you were lucky, have seen on the news, in the previous month, sometime. But it'll be a very sophisticated analysis that's worth reading. And it's not, of course, necessarily written for a particularly technical audience, though having read my book would tend to help. But it's written, something that will teach everyone, however good you are at biology, however much you know, it will teach you something. And we think this is very important. For those of you who haven't read my book, well, it's totally right, really, because it came out 3 years ago now. So do get hold of it if you haven't. The reason I mentioned the paperback edition is because it's got an extra chapter, and the reason it's got an extra chapter is because it came out 1 year later, 12 months later than the hardback edition. And in those 12 months, so much progress was made, all of the various areas that are covered in the book, all of the various components of SENS, that we just had no choice. We just had to write a whole new chapter just to cover 1 year of progress. So that's pretty good. And actually, for the rest of my remarks, I'm going to lean on that, because I felt, I want to tell you lots of new stuff. And the best way for me to do that is not to try to be structured about it, but just to throw out a few tidbits to titillate you. Everything I'm going to say in the rest of my remarks came out in the last month or two, all right? And some of them we've talked about on the website, on the blog, so obviously that's important. But also this will encourage you to look around and generally pay attention to what's going on. I'm not going to talk very much about the sorts of things you all already have heard about. So here's a nice example of something that I bet a lot of you have heard about. We've got a lot of press. A group in Winston-Salem run by Tony Atala, who is one of the world leaders in tissue engineering and who is a close colleague of ours. He's on our research advisory board and associate editor on my journal and so on. Released some very interesting work showing how to use something called organ printing, which has been around for a little while in an early stage, in a way that really works on a whole organism. In other words, you can actually take an injured mouse and rejuvenate its tissue, in particular rejuvenate its skin, using this technology. So this is a big breakthrough. It's got a lot of press, so it's not the sort of thing that we tend to focus on promoting. This is another sort of thing we don't tend to focus on promoting. This is a very good paper, I'm not saying this, it's a very good and solid piece of work. It's highly technical, and it's a pretty niche advance. It's a report on the discovery of one little extra mechanistic detail in how cell senescence works. You wouldn't know that if you'd come across this, which some of you may have, because this is a report on it. You see here, science is... I should emphasize something. This is not your fucking Daily Mail. This is the Financial Times. Right? It's completely amazing the way that these things get distorted. And of course, this pisses me off to a certain degree, because of course my work is distorted in the opposite direction. But there you go. I mean, this is what happens. So we don't talk about that on our blog either. We talk about things like this. So here's something that looks, from its title, to be, again, really technical, not very interesting, not very important. That's why you need people like me and Michael and my other research people, because we understand what this really means. This paper turns out to be an extremely important breakthrough in showing how we can use the concept of induced pluripotency much more effectively in the future than we can yet. So most of you, I'm sure, are familiar with the concept of induced pluripotency, the technology that was developed about 3 years ago now by a group in Japan, and is now being used by everyone under the sun in stem cell biology to take a fully differentiated cell and restore it, de-differentiate it back to an embryonic stem cell-like state, from which it can be used in a whole variety of different ways, just like regular embryonic stem cells. Now the thing is that an awful lot of things have been discovered about iPS cells, induced pluripotent stem cells, that show that they're not quite the real McCoy. There's a lot of little subtle things that we don't like about the way these cells behave. So we've got a lot more to discover about how this works. But exactly how much we have to discover is not clear. A lot of people have been worrying that there's an upper limit to how good these cells can be, however good we get at it. This paper is a real breakthrough paper showing that that's not true. What we have here is the demonstration that you can use exactly the extract from cells that are already in an embryonic state, and which have therefore a whole bunch of components that we haven't yet quite identified, and the efficacy, both the fidelity and the speed with which you can de-differentiate a genome, a nucleus in other words, from a differentiated cell, is just off the scale faster than what happens with the regular IPS technology. So this is a massive breakthrough, and it didn't get anything like the press that it ought to have got, because it really needs to be understood that this is where we can potentially get to just by refining the cocktail that we give to differentiated cells to de-differentiate them. This is the sort of thing that really defines the field now, and people like you, people who understand the potential of future biomedical technology, need to be told about this just as professional biologists need to be told about it, so that you guys can help to contribute to promoting research on this, to getting resources allocated to it and so on. Here's another example. So it turns out that if you take stem cells in cell culture, you get them out of the body, and you'd like to use them therapeutically, you've generally got to make a lot more stem cells. You've got to expand them up in the laboratory. Now, some cells that we use in the laboratory are very easy to expand. You can just grow them, and subject to telomere shortening, which obviously we do know how to fix, they don't change in the way very much in how they behave after they've been subject to a lot of cell divisions. But stem cells, by and large, that's not true. Stem cells lose their stemness, and therefore it doesn't matter how much you expand them, they're not very useful anymore for the purposes that you hope they would be used for. This is a discovery that shows how to fix that. What you've got to basically do is overexpress one particular cell adhesion molecule that gets cells to talk to each other better. And again, this got no publicity at all. This is a major breakthrough. It's a major breakthrough in something that sounds fairly technical, but it makes a hell of a lot of difference to the ability of people to actually apply stem cells in the clinic. And that's true even for current stem cell therapies, let alone future ones. Here's another thing. A lot of people probably know here that the first area that I got interested in, well over 10 years ago now, but more like 15 years ago, was the problem of mitochondrial mutations. Mutations in this special part of the DNA that's not in the nucleus at all, it's in the mitochondria. There's not very much of it only encodes 13 proteins. I'd really like those proteins not to be there, or at least to be encoded by nuclear genes as well, so that if you got this accumulation of mutations in the mitochondria, it wouldn't matter. And we've been beavering away on this. There was a big breakthrough a few years ago, which we have helped to contribute to and continue to do so, which showed how to overcome the big problem of putting these genes in the nucleus, namely the translocation of the encoded proteins back into the mitochondrion. That's sort of solved, but I want to emphasize that the devil is in the detail here. There are 13 of these proteins. That means we've got to get them all working at the same time in order to have a really comprehensive therapy. And even if you can do each one of these things individually with pretty good reliability, you're still not necessarily going to have solved the whole problem, because little technical interactions that you weren't expecting are going to happen. So it's got to be important to keep an open mind about how to improve on this. And this is a paper that makes a big contribution to that. It shows that you can make a very, very slight change, just one amino acid change to one of these proteins, which happens to be a reasonably tricky one, more tricky than a lot of them. and that's enough. So this is an example of the versatility that we're able to apply to what has historically been considered to be a virtually intractable problem.